Okay, we're on to part two, and part two is what to do with your blast results. And so here are my sample blast results. And you can see right away I've got a problem. And so we're going to walk through how you deal with each kind of problem that may come up. And I think I've got most of them somewhere in my data set, so that's good. Okay, so these things are all going to be in the order that we put them in. And if you want to see what that order is, it is back on our Open Reading Frame Finder page, which is hiding right back here. There we go. And so first I'm looking at Open Reading Frame 1. So it's going in the forward direction. It's starting right there. It's partially overlapping Open Reading Frame 4. So it may not even be real. Open Reading Frame 4 is bigger, and bigger usually means better in open reading frames anyway. You're less likely to get a big gene occurring by chance. Something that looks like a small gene can just happen randomly because there's only 4 bases, A, C, G, and T, and eventually you'll get an A and a T and a G next to each other that looks like the start, and eventually you'll get a stop at the end of that. And the stuff that's in the middle may not actually be important, and the bacteria may not actually make much of it. But here we go. Let's look at this. And so we'll put this screen capture down for a little late. Use this later. We're looking at Open Reading Frame 1 first, and we're going in order. Okay, and so it says Open Reading Frame 1 is 205 amino acids. That's helpful. And it also has a little star next to it, and it says no significant similarity found, which means that the thing that we found, this potential protein that we put in, is something that has not been annotated. People may or may not have seen it, but nobody has bothered to actually take the time to write down what this thing is. Nobody's ever worked on it. Nobody's ever reported it. Probably when you encounter something like this, probably it's just junk. It's just sort of the white noise of biology. There's a chance it's something totally new and really interesting, but you'd have to do a whole bunch of experiments to figure out whether that is the case. And that's not what this project is about. This is about taking whatever bioinformatics can give us. And bioinformatics is basically attaching information, like human understandable information, to a sequence, which just looks like gibberish. I mean, look at our sequence over here, ACGT. I can't make any sense of that. But with these programs, yeah, we can make a lot of sense of it. So Open Reading Frame 1 didn't come back with anything. That's fine. Sometimes they do, sometimes they don't. And look for the little star there, and sometimes the text will be grayed out. It'll be like a lighter gray font than this black font if it found nothing. Let's look at a different one. So there we go. Okay, and this looks like a much better one. So Open Reading Frame 2 is 327 amino acids. Great, that's fine. That's a stat. You can write that down. And then down here, this is this is the meat. So this is what you want to put onto your map, the data that you're going to need. So here's our little map here, and so we're looking at Open Reading Frame 2, and I see Open Reading Frame 2 is right there. There it is. And so that looks great. And it's saying that Open Reading Frame 2 is similar to a lot of things that already have names, and that is wonderful. That's what we want. So the first part of this thing is the name of whatever the protein is. And I'm looking at that and I see it ends in ASE, so it's some kind of enzyme, and so that's great. And so what does this enzyme do? Well, we're going to read it backwards, starting at the ASE. So it says it's a transferase, which means it moves some molecule onto some other molecule. It's probably adding a molecule to another thing. And so what's the molecule it's adding? Well, you look back one step more, and it's a sort of formate molecule, so a short carbon chain that is going to be added on to something. And so what are we going to add it to? Well, we're going to add it to methionine tRNA. So methionine is just an adjective version of methionine, the amino acid, and tRNA is the actual transfer RNA that is going to take care of putting methionine into a protein. So somewhere on this thing, it's going to have an anti-codon that binds the codon for methionine. And so the codon for methionine is ATG, and so this will have the opposite codon. And boom, it'll stick to that, and the ribosome will take off the methionine and stick it on there. And so this is a formal transferase, so it puts a formate group onto methionine, so it's going to make a thing called formal methionine. And that is actually how every bacterial protein starts. They don't start with methionine, they start with formal methionine. It's just a different version of the thing. Got an extra molecule on it, but it serves basically the same purpose. And so for our purposes, we just have to document this. We have to annotate this. Okay, so we got a formal transferase, a methionine tRNA, formal transferase enzyme, and ours is most like one that comes from this bacterium. And so this is the genus here, Ralstonia, and this is the species Solanacearum. And so a genus is like a bigger group of things like there's Homo sapiens, those are people, and there's Homo neanderthalensis. These are Neanderthal cavemen that don't exist anymore. They were a different species, but they're in the same genus, the genus Homo, which we are in as well. And so this is a thing in genus Ralstonia, species Solanacearum. Great, awesome. And that's the one I was expecting that you would find. This is what we got these samples from Colombia, and they were damaging banana crops. And you never know exactly for sure what a bacterium is until you sequence its genome. But this is at least one piece of data that says it probably was Ralstonia like we thought. Now, before we can do anything with this information, we got to decide, do we believe this information or not? And so we got to go over here, and these are the stats that are really going to help us. So the big stat that we really want is the E value, and E in this case means chance of error, like chance that it's not actually a formal transferase from Ralstonia solanacearum, chance that we're wrong about that. And the number it's giving us is 0, which actually means it's less than 10 to the minus 180th power, which is a tiny number, and so it might as well be 0. And so this is saying you have a 0% chance you're wrong, basically. And I like those odds. Yeah, so that means that probably we do have a formal transferase. And whatever our bacterium is, it's probably pretty close to Ralstonia solanacearum. So that's the top hit statistically, and then it has the other hits going down here. These are also other possible hits. And if you notice, they look real similar because lots of different people have sequenced Ralstonia genomes. And every time they do, they seem to find a methionine formal transferase because probably it's real important. If this is the thing that makes the tRNA that starts every single protein Ralstonia makes, then yeah, you're going to find it every time you really look at the genome carefully. So here we go far enough down the list, and we've got one that is pretty close, 97% identical. And Ralstonia, what is that? Pseudo-Solanacearum. Okay, so something real close to Solanacearum. And then we've got down here, these people were willing to say it was a Ralstonia, but they were like, it's probably something weird about it. We don't want to say it's Solanacearum for sure, etc. And you got species that are just numbered down here. And you got Piketty down here, Ralstonia Piketty, and Insidiosa, etc., etc., etc. And if you get on far enough, you're past all the matches that are super close, like Ralstonia, and you're down into matches to other kinds of bacteria that are related to Ralstonia. And so Ralstonia is in this group called the Burkholderaceae, and so is Cupriavitis, so is Pandoria, so are all of these things. And so you've got a gene that seems to be conserved in a whole group of bacteria. And that's what we're showing here. So that one's easy. Now let's go and find one that's a little trickier. And we may have to look through a couple of them until we find it. So that's another very nice one. And just as a little check, so what's this thing? This is a methyl transferase. And so it's been named RSMB by somebody who found it. And so, that's like a shorthand for this, but we want to know what this is because, honestly, nobody knows all their bacterial genes. This would only mean something to somebody working in the field of Ralstonia biology, and even then, it would only make sense to a couple of them. So it is a transferase, it is a methyl transferase, so we're going to transfer a methyl group instead of a 4-mel group. And what are we going to transfer it to? Well, the 16S ribosomal RNA, one of the RNAs that make up the ribosome, has a cytosine, a C nucleotide, at position 967. It's a big, long RNA. And on that thing, at the 5th carbon, we're going to add a methyl group with this particular enzyme. So it's super specific. And actually, before we could even look at that, we should go over here and say, what's our E value, our chance of error? And our chance of error is, again, 0, and so I'm confident. We don't have to talk about all the rest of these. As long as we've got a good first hit that makes sense, boom, we can just stick with that. And it is 100% identical over 100% of the protein. That's what query cover means. And so I think that's something you can believe in. So, all right, open reading frame 2 and 3 were easy. And now let's see, 4. Okay, 4 looks okay. Fine, so we got a histidine kinase. Great. It's going to attach a phosphate group to a histidine of some particular protein. And then right after that, I think we have our other part of the two-component system. So there's usually a histidine kinase and usually a regulator. These two things go together, and they usually occur right next to each other in the genome, and they're both turned on at the same time. So you may find something similar in your section. You may not. Yeah, bacteria have a lot of genes, several thousand genes. So there's always a lot to look at. Okay, let's look at another one. Great. So this is a thing called a lyce-E, and so that's another one of those names that'll make sense to Ralstonia people. So it's part of the lyce-E family, and it's a translocator. And I don't really know anything about that, but we can sure Google it. LYSE translocator. Yes, that thing. And there we go. Aha. So, there we go. This family of proteins consists of hypothetical proteins and an L-lysine exporter. So L is the normal version for amino acids. D is the other version, like the right-handed version. And those are weird, and you usually don't get those incorporated in proteins. There's only a couple cases, and it takes some really weird machinery to put a D amino acid in. Anyway, okay, lyce from Corynebacterium, fine, is proposed to be the first of a novel family of translocators. So a translocator is something that moves a thing from one side of the membrane to another, and it looks like that's what this does. It crosses the membrane 6 times, and it is going to excrete excess lysine if you get too much lysine building up inside the bacterium. So, okay, that's neat. Yeah, cool. And so we found one of those. It's yeah, it's a special protein dedicated to getting rid of a particular thing. So however this bacterium lives its life, it must end up generating a little bit too much lysine. Otherwise, it probably wouldn't have any use for one of these things. So great, we got another gene here. And all this information that we're finding out about these proteins is very good for writing up that section. Zinc Metalloprotease, so the A's, okay, that tells us it's an enzyme. HTPX, that's just what somebody has called it, but wouldn't necessarily mean anything to you or I. So this is a protease, which means it's a thing that cuts protein. And it's a metalloprotease, so it's a thing that uses a metal ion to cut a protein. And what metal ion does it use? Well, it says it's a zinc metalloprotease. So zinc is a metal, and zinc ions they have a 2+ charge, are often held by proteins, and they use these in various ways. This one is using a zinc as kind of the battering ram to break proteins. So great, that's an HTPX type zinc metalloprotease. Now we're going to eventually get to something that is a little bit unusual, I think. Here we go. Okay, so this is one of the weird ones. So, all right, great. So we've got these things. They're saying it's been found before in Ralstonia solanacearum, and that is a subspecies, P082, like an isolate of Ralstonia solanacearum. You don't have to worry about that. If it's in this species, let's consider it all to be the same thing. So it is a DUF4390 domain-containing protein, and what that means is we don't really know. And this thing, probable signal peptide, proline-rich protein, these are people basically throwing up their hands. When they've got a protein, seems to be real, seems to keep occurring over and over again. Yeah, look at all the times it pops up. But we really have no idea what this thing does. So DUF stands for Domain of Unknown Function. And there's a big list of them, and this is number 4390 on that list. Occurs in different places, and until somebody works on it and figures out what it is, we are not going to know what it is. And so if you see a DUF there, or you see, here we go, conserved exported protein of unknown function, or you see hypothetical protein, something like that, all those are signs to you that we're not going to be able to say much about this protein. So you're going to annotate it. You're going to say that it is a DUF4390 containing protein, or that it's a possible signal peptide, proline-rich protein, but you're not going to be able to say any more about it. So this is one of those cases where you found something kind of new, and we're actually out ahead of the field. We know about this thing, and nobody else has really figured out what it does. So great. Let's just keep going through our list. So that was open reading frame 8, and open reading frame 8 on our chart. Here, let's drag it up here. Is there it is. Open reading frame 8. So yeah, okay. And yeah, interesting. So, fine. Now let's go. Here's open reading frame 9. Whoa, okay. Open reading frame 9. The trash train continues to pull into the station. Wow, that's really something. That's a lot of junk. Okay, so before we always had these nice red hits. Red hits mean really good, confident hits. Now we're getting purple, blue, green, black hits. These all mean, grain of salt. And the cooler the color is as you get toward this end, the more salt you're going to put on there until you finally get to the point where you say, I don't believe that. That doesn't even, that's not well supported. And so look at these E values. At the top, yeah, we can probably believe that. That may well be correct. But down here at the bottom, wow, E values of 4, of 9, of 9.9. Yeah, no way you want to believe those. Those are terrible E values. And even these are kind of, You can generally believe anything that is smaller than 1e minus O2, which is a 1% chance of an error. I'm willing to accept that much, and that's just an arbitrary cutoff. Everybody picks their own cutoff. But here, let's see what it is. So it's a putative protein tyrosine phosphatase. Interesting. Could be. Or an uncharacterized protein. Yeah, doesn't say much. Uncharacterized. Doesn't say much. Predicted protein tyrosine phosphatase from a different kind of bacteria. So yeah, okay. And so this doesn't mean that we have found a mycobacterium. It just means that this Mycobacterium avium, which is kind of like tuberculosis, but it gets into birds. It has the same gene that our bacteria have. So it may be a widespread gene. It may help it live in a particular place or do a particular task. And it'll be all over the place. You'll find lots of bacteria in the environment that have this thing. Yeah, so I think that's what you would call it. You'd say it's a putative protein tyrosine phosphatase. Okay, let's look at these last three really quickly. And so, boom, now we got another one that is an uncharacterized protein. This one says it could be an aldo keto reductase, which, great, it could, but when I look at that E value, 2.1, so that means there's 2 chances that I'm wrong for every 1 chance that I'm right, and I don't like those odds. I don't buy it. The one that I can believe right here is an uncharacterized protein. And if it's uncharacterized, that just means nobody's ever worked on it yet. And so you have found a copy of this thing from Burkholderia, which is a related bacterium. And nobody knows what it does in either of those cases. So you can list it as an uncharacterized protein, say it's got a great E value, but we can say absolutely nothing else about this. And so let's just click through the last couple. There's another one, terrible. We got a good E value, so we can believe it, but it's an uncharacterized protein or it's a hypothetical protein. Either way, we can't say anything about it, and so we can just note it, note where it is, and move on. Same thing there, hypothetical protein. And, an ABC transporter would be neat. These are the multi-drug resistance proteins, but with an E value of 8.2, I am not going to make that call. I do not believe that. And you shouldn't either. This is where you learn how to judge these things based on the numbers. And here's the last really trashy result, and so mine is probably a hypothetical protein or an uncharacterized protein, and everything else has bad E values that I do not like. And so, yeah, that's fine. We're going to note it, note where it is, and move on. And note the E value and what it's a match for. So the other point to make here is that this is an E value saying we think it is this kind of thing, but what it's actually saying is it is exactly like this particular sequence. And so this is the actual sequence that yours resembles. And we can click it, and we can go there, and we can read whatever people have bothered to put on this one. So this will tell you who actually found this thing, the authors of the paper, and it looks like they sequenced some bacteria, and we're getting ready to publish them. or maybe did publish them. Yeah, I think that's from a while ago. Yeah. And so it's saying, well, here it is. This is the protein that's here, something like. And it doesn't really say anything else there except that it's a hypothetical protein, but boom, yeah. So this is the type of protein, like a hypothetical protein or a methyl transferase or a formal transferase. That's the name of a group of proteins. Then you'll find those in different organisms. And then over here, this is the actual thing that it matches most closely. So that's the accession number, and that is unique. If you type in EXI88400, it will take you to this thing every single time, which is nice. Yeah. Okay, so that's the end of that section, how to interpret a blast result. And with that, you've now got something to write about. You've got all these interesting terms, and essentially, the person who's in charge of this section is going to be going and looking up what these terms mean and how do they work together. So 16S ribosomal RNA, cytosine methyl transferase, RSMB. How does that thing, what does that thing mean? And you're just going to pop that into Google and you're going to search for papers on it and you're going to find some. Guarantee it. Okay, that's the end of this section. Thank you very much. See you on the next section.